I hope you are doing well. Today's lecture is about tumor immunology and then practical ASO test. What do you know of tumor immunology? Whenever you hear tumor immunology, what comes to your mind?
Like cancer.
Cancer. Please?
Yes, cancer cells.
Cancer cells.
Infections.
When you hear cancer immunology, what comes to your mind?
How our
What
immune
do you
system
think?
deal with cancer cells?
How immune system deals with cancer cells?
Response the immunity against the tumor.
Response of immunity against tumor. Yes. What else? If you want to define
Transform cells and convert.
transform cells,
Yes.
conversion of normal cells towards cancer cells. Yes. And if you want to define in one sentence tumor, what would you say? Definition of tumor or cancer.
Normality growth to cells.
Abnormal
Normality growth
abnormal growth
to cells.
of the cells. Abnormal growth of the cells. What else?
The change in the genome of the normal cell because maybe the virus after that convert to the cancer cell.
Change in the genes of the cell, genome or genes of the cell, which they are called mutation. Do you mean?
Or ionization.
Mutation in the cells that finally leads to cancer. What else? What else? If we want to define, or if we have a short definition of cancer, we can say uncontrolled proliferation or multiplication of the cells. Uncontrolled proliferation. All cells of the body have normal proliferation, normal multiplication, but under some circumstances we have abnormal, uncontrolled, higher rate of proliferation. So it's called tumor or cancer. What causes cancer in normal? I mean, what is the difference between normal and cancer? That in normal cell we have controlled proliferation. Whenever it is needed, we have proliferation. But whenever there is no need to proliferation, we do not have proliferation. But in cancer is not so. Uncontrolled proliferation we have in cancer.
No
What's
homeostasis.
the differences between?
The cancer cell, no homeostasis.
No homeostasis. There is no homeostasis in cancer cell. What's the reason that we do not have?
Generate virus for point. Division infection, lifespan, and movement.
Genes that are controlling
Yes.
these aspects of the cell.
Having a mutation to genes that control the proliferation of
Thank you.
the cells that
Thank
happen
you.
by UV or ionization or any different X-ray effect.
So the main difference, thank you. The main difference between normal and abnormal, or in this lecture, normal and cancerous cell, is uncontrolled proliferation. And main reason of uncontrolled proliferation is mutation. Mutation, everything that happens for cancer is a result of mutation. Everything that you will say in the next slide. Everything that happens in cancer is indeed a result of mutation. So, as a result of mutation, we have uncontrolled proliferation of the cells. And do mutation in all genes of the body, mutations in all genes of the body can cause cancer?
No.
What?
No.
Let me ask my question again. Do mutations in all genes of the body can cause cancer? Or in another word, all mutations, all possible mutations can cause cancer?
No, doctor.
No.
No.
Why?
Just
You said my answer.
the the changing by virus can, but changing by virus can cause the tumor cell.
I didn't understand. Can you explain it in another way?
The mutation.
Mutation in, my question is this. Which type of mutation can cause cancer? The main question is this. Do all mutations can cause cancer? Of course not. So, mutation in which genes of the body, in which type of genes of the body can cause cancer? DNA, whenever we say mutation, indeed, we mean alteration in DNA sequence. Any alteration in DNA sequence is called mutation. So, mutation in which type of the cell genes can cause cancer? On what do you mean by oncogene? Genes responsible for growth factors. Growth factors are the type of oncogene indeed, or growth factor receptors. What else? It could be right, but
Any differentiation in the sequence of gene that like
it is called
thymine-
mutation.
thymine.
Any alteration in the sequence of the genes is called mutation. Alteration of the DNA is mutation. So it is possible that we have alteration in DNA or mutation of the genes of all genes of the body, about 20,000 different protein genes. If we have mutation in any genes of the body, it is possible to have a cancer? Of course not.
It can't change the shape or specific.
You said my answer in my previous question. Genes, mutation in the genes that they control the proliferation of the cell. Oncogenes are a type of genes that they promote proliferation. So mutation in genes that they control the proliferation, or it's better to say they control cell cycle. Do you remember in biology?
Yes.
Cell cycle. We have several genes that they control cell cycle. Some of them are a promoter of cell cycle.
Yes.
They encourage cell cycle. Some of them prevent cell cycle. We have both of them. And mutation in all of them, oncogenes are a type of genes or a series of genes that they promote cell proliferation. And some other genes prevent cell proliferation, or it's better to say they make a stop in cell cycle. And mutation in these genes can cause uncontrolled proliferation. And the second the second type genes are called tumor suppressor genes, genes that make a stop in cell cycle. So until now we talked about tumor definition, mutation that are the different mutations that are the main reason of cancer formation, and types of the mutation that indeed they can cause mutation. We talked about types of the mutation. I mean types of the mutation in the types of gene. We might have insertional mutation, deletion mutation, any type of mutation that can induce, that can change the sequence of genes that are controlling cell cycle. Besides, mutation in the genes that control cell survival and death. So any genes that control cell survival and death, like apoptosis gene. Do you remember? Apoptosis genes, we have several genes involved in apoptosis, they product indeed. Any mutation in the genes of apoptosis, and besides, mutation in the genes that control cell cycle can cause cancer. So cancer is uncontrolled proliferation, and mutation in genes that control proliferation and cell survival. What causes mutation?
Virus.
Viral infection. What else? Radiation.
Ultraviolet. Yes.
Ultraviolet. Yes.
Physiology.
Chemical. So you know they are called carcinogens. Viral infections, chemical agents, physical agents like UV, and many others. Radiation. They are the main cause of mutation. So exposure to carcinogens, constant exposure to carcinogens can induce mutation in the genes, several genes. And whenever we have mutation in the genes that are controlling cell cycle and cell death and survival, it is possible to have a cancer, uncontrolled proliferation. This is a very short summary of cancer formation, important mutation, and main cause of mutation induction. Clear? Yes. So, we talked about very shortly about some background on cancer, and then I'll talk about the main topic of this lecture is to understand how immune system fight against cancer, how immune system deal with cancer, and then how cancer cells can escape from immune system. Immune system has evolved to detect cancer and eliminate it, and besides, cancers have gained some mechanisms to escape from immune system, to suppress immune system in order to proliferate in an uncontrolled way. So, and then, in summary, if I have time, I'll talk about immunotherapy against cancer. Let me skip the key terms. You know most of them, and I'll talk about some of them in the slides. We do not have enough time to talk about all of them. You know most of them, just as a reminder. So, cancer is the second cause of death worldwide after cardiovascular disease. So it's very important how to deal with cancer, how to treat cancer, how to diagnose cancer, how to prevent cancer. All of them are very important. Prevention of cancer as much as possible. Detection or diagnosis of cancer as soon as possible. And using of anti-cancer therapy as soon as possible and as efficient as possible. The efficiency of different cancer therapies are different. And the aim of the recent decades therapies against cancer, immunotherapies, are to increase the efficiency of cancer treatment. And almost half of men and 1 in 3 of women will have cancer. Half of men and 1 in 3 of women, besides 20 to 25% of us. 20 to 25% of us will die of cancer. Us, ourselves, and our families, our relatives. So it is very important. It is very important to find a way to prevent, as I said, to prevent cancer, to diagnose cancer, to treat cancer, in order to help the families that whenever we have one person died of cancer, several persons around him, his or her relatives, family members, friends are involved emotionally. And healthcare systems are involved. Most of the expense of the healthcare system is devoted to cancer detection, cancer prevention, cancer treatment. So it's very important, and it's our responsibility to find a way in all steps of cancer prevention, detection, and treatment. So, we are going to talk about immune response against cancer, how immune system fight with cancer. There is a main challenge for immune system. There is a very important challenge for immune system to fight with cancer. What's the challenge, do you think?
Identify the cell if it's normal or abnormal.
Why the identification of normal from abnormal is a challenge for immune system?
Because it looks like other normal cells.
Yes, cancer cells are derived from normal cells, and most of their antigens are normal self-antigens.
Yes.
So discrimination between cancer cell and normal cell is a challenge for immune system.
We're using chemical and ultra-wise therapy to make it move faster. So immune system identify this is cells have to kill it, so it's kill it. That's why we have the diarrhea and hair loss because our
Chemical agents.
yes because
chemotherapy.
because these cells has movement a lot.
Yes. So, thank you. So, immune system, there is a challenge for immune system to discriminate between cancer and normal. Cancerous cells and normal cells. It's because, as I've told you, and as you know, the cancer cells, almost all of them, not all of them, antigens of the cancers are similar to normal cells. And immune system has evolved. Do you remember? I have told you that immune system has evolved to discriminate between self and non-self. Self cells, self antigens, and more exactly, self epitopes from non-self epitopes, microorganisms, several type of microorganisms. And besides, immune system has evolved.
يقدر محمد حسن وين؟ بس تفضل.
Okay, let's go on. Immune system has evolved to discriminate self from non-self. And besides, immune system, please, has evolved to discriminate between normal cell, normal antigens, and abnormal antigens. Do you remember? Whenever immune system innate immunity, first line of immune defense, whenever immune system wants to discriminate between self and non-self, in innate immunity we have several types of PRR, do you remember? Pattern recognition receptors that they recognize, that they can distinguish PAM, pathogen associated molecular pattern.
Down.
Do you remember?
Yes.
We have several types of PAMs that they are recognized by several types of PRRs in innate immunity in order to discriminate between self and non-self for innate immunity. In acquired immunity, we have TCR and BCR. Besides, immune system has innate cells, have several types of PRR to bind, to distinguish, to recognize DAMs. Do you remember?
Yes.
Damage-associated molecular patterns. Whenever we say damage-associated molecular patterns, indeed we mean altered self, damaged self. Whenever we have any changes in the antigens of the body, immune system can discriminate between altered self and normal self. One main function of innate immunity is tissue repair. In order to immune system to help tissue repair, should discriminate between damage and normal. So there are several types of PRR to recognize DAMP, antigens derived from damaged or altered antigen. Is that clear?
Yes.
Several PRRs, PAMs for non-self, DAMPs for altered self. Discrimination between self and non-self, self and altered self. So in cancer, of course, we have some DAMP, or it's better to say, of course, we have some altered antigens, or altered expression of the antigens that immune system has evolved to discriminate, to distinguish them in order to discriminate between cancerous and normal cell, and to solve the challenge we talked about. Question? No? So, immune system in both innate and adaptive, has evolved to discriminate between cancerous altered antigen and normal cells. And based on this discrimination between altered abnormal cells and self-antigens, immune system, if detect an abnormal antigen, an altered antigen, tries to eliminate the antigen. So this is an ongoing process, a constant process inside the body, which is made by immune system to detect cancerous cells and to eliminate them. So detection and elimination of the cancerous cells is made by immune system constantly now that we are here. Our immune system always try to detect and eliminate cancerous cells. Is that clear? So detection of cancerous cells and elimination of them is an ongoing process which is called immunosurveillance of the cancer. Surveillance of the body against cancer by immune system. Immunosurveillance. So immunosurveillance is a very common term in oncology and immunology against tumor or tumor immunology. That means ongoing process of detection and elimination of the cancer. And in this lecture, I'm going to talk about the basics of detection, how immune system can detect and how immune system can eliminate the process of them. And then I'll talk about the mechanisms that cancer can escape from immune system. Question, comment? Whenever we say another term for cancerous cells is transformed cell. Transformed cells indeed are cancerous cells that their form has changed from normal to cancer. Transform, change in the form of the cells from normal. So whenever in the textbook, in the papers you say transformed cells, is another term for cancerous cells, tumorous cells. I talked about these concepts in summary. You can read them from the slides. Let me go to the there are some tables in the slides. There is no need to memorize them. They are only as an example. For example, viral infection that can cause cancer, or some genes that are involved, oncogenes, tumor suppressor genes, several types of genes, apoptotic genes, that mutation in all of them can cause cancer. But there is no need to memorize them. Okay. There is very important concept about cancer, that cancer formation occurs or cancer development is a multi-step process. And it occurs over time. It's very important concept about cancer. Cancer formation, cancer development occurs in a multi-step process over time. Sometimes it takes 15 or 20 years in order to have a cancer, from the first mutation to having a full cancer, an invasive cancer, I mean. So it takes several years, 15 years, 20 years. What do you think the reason of that? Why it takes several years in order to have an invasive cancer? From the first mutation, I mean cancer causative mutation.
Cancer causes mutation from the first cause of mutation. It's maybe doesn't happen in the application, or it's wait.
No, I'm talking about cancer causative mutation. I mean the mutation in the genes that I talked about it. Genes that are controlling proliferation and cell survival and death. From the first mutation until to have an invasive cancer, it takes several times. What's the reason?
It gets many mutations that when it's reached to the
Invasive.
yes, invasive genes that doesn't control the duplication of the cells.
Yes, what else? Did you get my question?
Yes.
Let me ask it again. For example, in one cell, we have a mutation, first mutation, in the genes that is controlling the proliferation or cell cycle. This is the first mutation in the cells. Mutation in the genes that control the cell proliferation. From this first mutation, for example, until to have an invasive cancer takes several years, 15 years or 20 years. What does this process take several years in order to have an invasive cancer? Did you get the question? We have one cell here. First mutation is caused, for example, as a result of UV exposure. We have one mutation in one gene that controls cell proliferation. But we do not have cancer yet. And it takes several years, 15 to 20 years, in order to have a tumor mass. What's the reason that this process takes several years? Did you get the question? So,
I think it's for growth the cells, growth the
growth
cells,
of the cells?
and make it ready to separation.
No.
Because the single mutation is not
Enough.
to make a cancer, so it is related to cells needs to the cell several cortical
Yes.
genes.
First reason is this. Mutation in one gene, even one gene that controls cell proliferation, is not enough to have cancer. Because cell cycle, cell death, cell survival, all of them are controlled by several genes. And mutation in one gene is not enough to have a cancer. This is the first reason of taking so much time to have a tumor mass or an invasive cancer.
I just said it.
We have another reason besides this. The action of immune system. Immune system tries to prevent cancer formation, but it takes time in order to, please, the second reason why having a tumor mass, having an invasive tumor takes several time from the first mutation is that immune system mechanisms try to prevent cancer formation. And it takes several years for cancer to develop several mechanisms. It takes, please, several years for cancer to develop mechanisms in order to be able to escape from immune system. At first, transformed cells, the first cell which is transformed is not able to evade from immune system. It takes several years to gain that capability. Is that clear?
Yes.
So the action of immune system and having one mutation is not enough to have cancer are the main reasons that's why forming of cancer takes several years. You will see the process by which immune system can fight with cancer and how cancer can evade from immune system. So the first mutation and everything's gaining new capability in cancer in order to evade from immune system, all of them is result of mutations. Over time, some of us right now might have susceptibility genes for cancer that we have inherited from our parents. But it doesn't mean that we have cancer. There is only susceptibility. There is need for other mutations over time with exposure to carcinogens. First mutation is not enough. Second mutation is sometimes several years exposure to carcinogens and induction of new mutation, new capability in the tumor, in order to have uncontrolled proliferation, besides in order to be able to escape from immune system. Clear? John. Yes.
What is this red yellow?
The red one,
Red,
this is
yes.
the mutation. A carcinogen, for example, a carcinogen that cause alteration in the DNA. For example, UV, asbestos, viral infection, several carcinogens that can cause DNA alteration or mutation. Another concept about cancer is that a cell population of a tumor mass, cell population, I'm talking about, please, here I'm talking about the cells of one tumor, not several tumors. The cells of a tumor mass in one person. The cells of a tumor mass in one person are genetically heterogeneous. It's because the rate of mutation in the genome of tumor cells is higher than normal cells. So there is a higher probability to have mutation. Salam alaikum. Welcome here, sir. Sorry. So, this, as I told you, the rate of mutation in cancerous cell is higher than normal cell. As the rate of mutation is higher than normal cell, so it is possible to have a heterogeneous population. And this is another challenge for immune system to fight with cancer. For example, we have a mutation here that caused a tumor mass, this population, but there is probability to have another mutation. We have a second mutation and arising of new population. Again, new mutation, arising of new population, and it's an ongoing process. It occurs all the time in tumor cells. New mutation, new population. New mutation and arising of new population. And as I told you, it's a challenge for immune system. It's because, for example, we try to develop an immunotherapy against these populations of the cell. And our strategy can kill all of the population. But there is a single cell with new mutation that can resist against our therapy. Okay? Any resistance in cancerous cell is as a result of mutation. So we try to develop a strategy of immunotherapy. And we try to kill all of this population. But again, we have a single cell with new mutation, with new capability that enables that cell to resist to immunotherapy. So it will induce, it would be the reason for tumor relapse after several months and sometimes several years. And anything inside the tumors, you can see in previous slides and here, that anything in cancer is result of mutation. It is said that cancer genome is unstable compared to normal genome. And the rate, it means that the rate of mutation is much more higher than normal cell. Question, comment? We have, so, now I'm going to talk about tumor antigens. Whenever we talk about antigens of anything, tumor, transplantation, microorganisms, now tumor or cancerous cells, indeed, we are going to talk about how immune system can detect the cancer, can discriminate the cancer from normal cell.
Some antigens it is on the cancer cells, so immune system it is can identify and found the cancer cell from the normal cells.
Yes.
Like tumor specific.
Specific antigens. Thank you. Yes. So whenever we are going to we are talking about tumor antigens. Indeed, we want to tell how immune system can discriminate self from cancer, from cancerous cells. If immune system want to discriminate between normal and cancerous cells, immune system should see differences between normal and cancerous cells. And these differences indeed are antigens of the tumor, that they are different in shape or the pattern of expression. Two kinds of differences. Some of them are in the shape of the antigens, another indeed in the pattern of expression of the antigens. You will see in the next slides. Generally, tumor antigens categorized in two main groups. The first groups, yes, tumor-specific antigens. As the name implies, they are the antigens that only are expressed on the tumor cells, not normal cells. We have them only on the cancer cells. So whenever we have in normal cells, we do not have such an antigen, and we have them only on cancerous cells, so immune system can discriminate cancer from normal. There is a difference between normal and cancer. Tumor-specific antigens. For example, antigens that are product of new mutation. I've told you, there is probability of having mutation in any genes of the cancerous cells. So any mutation can produce a protein that we have a mutation or change in the sequence of amino acid of that protein. That it is called new antigen for the tumors. Or sometimes some viral antigens, oncogenic viral antigens, are indeed tumor-specific antigens. For example, whenever we have HPV infection, hepatitis B virus infection, HPV can cause liver cancer. HPV, besides, only infect liver cells. So, whenever we have only the HPV antigens in the liver cell and we have cancer in the liver cell, it means that the antigens of the HPV are expressed by liver cell and T cell can detect them. So we have a cancerous cells, liver cancerous cells, that they are expressing viral antigens with their MHC class 1. Do you remember? We have MHC class 1 on the surface of all nucleated cells. And oncogenic viral infections, that now HPV has infected a liver cell and liver cell is expressing the antigens of the HPV. And this HPV antigen is only on the liver cell, not other cells. We do not have HPV antigen on the kidney cells. We do not have HPV antigen on the heart cells, on the muscle cells. We have them only on the liver cells, that now it is cancerous. So immune system again can discriminate between cancer and normal cell. Clear? Another type of tumors antigens is called tumor-associated antigens. Tumor-associated antigens are antigens that are expressed on normal and cancerous cell as well, in both of them. But there is difference in the pattern of expression. For example, in the tumor cells, we have a higher rate of their expression. In normal cell, we have lower expression of them. For example, in a normal gastric cell, we have 100 copy number of one growth factor receptor. It's an example only. 100 copy number of one growth factor we have on the gastric cells, in normal gastric cells. But whenever the gastric cells transform to cancerous cell, we have thousands or sometimes millions of that growth factor. So there is a difference. Lower expression, higher expression. And again, a differences between normal and cancerous cell. And immune system can detect this difference. Clear? So, knowing of this, what's the importance of knowing of these antigens? Is that, I mean from clinical perspective, what's the importance of having knowledge about tumor antigens? Is that only to pass the exam? No, of course. They are useful in clinic.
The immune system can identify the tumor cells from the normal cells.
Yes,
So this,
the first application of them for immune system is discrimination between self and non-self. But we can use these antigens in clinic. What do you think?
Surgical removal?
No. immunotherapy. We can target cancerous cells with immunotherapy strategies if we find a tumor antigen. What else? So first one is using them as a target for immunotherapy, using of tumor antigens. Another application of tumor antigens in clinic. No. Transplantation, no.
Indicate cancer development.
Do you mean diagnosis or detection of the cancer in the clinic?
Yeah,
Yes,
Yes,
because
it's
the immune
Yes,
cells.
we can use tumor antigens to diagnose the cancer, and they are used in the clinic now. Several tumor antigens are used as a tumor marker to diagnose the cancer in the clinical laboratory. So they are important. It's because we can use them as a diagnostic marker to diagnose, as I told you, several tumor markers are detected in the clinical laboratory right now for several types of cancer. Besides, we can use them to monitor cancer therapy, cancer treatment, if the efficiency of the cancer was high or low. Besides, we can use them as a prognostic marker and use them as a therapeutic target. They are targets of immunotherapy. That immunotherapy strategies in recent decades has revolutionized the treatment of cancer for several cancers. Question, comment? So, I'm going to talk about several types of tumor antigens from now on. That some of them are tumor specific, some others are tumor associated. The first tumor antigen is new antigen. As the name implies, new antigens. They are the product of new mutation, that we have the mutation only in the cancer. So, immune system can use them to discriminate between normal and cancerous cells. You can see here a normal cell. We have a normal, this is a normal cell, and one protein as an example inside the cytosol of normal cell, that is, and the cell, this is MHC. Do you remember all nucleated cells? Please, all nucleated cells are expressing MHC class 1 all the time, constantly, and they are expressing self-antigens. So, this is a normal cell, these are MHC, and these are the normal antigens derived from normal protein. Normal protein, normal epitopes, a small part of antigen, binding to self-MHC. So we do not have immune response here. Everything is normal. But in tumor cell, as a result of mutation, we have a new epitope. Mutation means alteration in the DNA that finally lead to having a different sequence of amino acid of that protein. So new epitope. New immune system can recognize new epitope. New epitope expression on the MHC, so T cells can discriminate between normal and cancerous cells. Normal epitope, new epitope, as a result of mutation. Clear?
Yes.
Question? Another antigen of the tumor is oncogenic viral antigens. I explain you. For example, this is a liver cell which is infected with, for example, hepatitis B virus. And you know that whenever a virus infects a cell, the proteins of the virus will be expressed by the target cell. And whenever we have proteins of the oncogenic viral inside the cells, they are expressed on the cleft of HLA1. HLA1, it's very important to remember that. HLA1 all the time samples proteins of the inside of the cell and express them to the T cells. It doesn't matter what kind of antigens are inside the cell. They are viral antigens or normal antigens or new antigens. In previous one, MHC class 1 was expressing new antigens. Here, MHC class 1 is expressing viral antigen. And this viral antigen is considered non-self for T cell. And this viral antigen is only expressed on the cancer, liver cancer cell, for example. not normal cell. Normal cells do not have HPV. Only cancer cells have HPV. Both of them, oncogenic viral antigens and neoantigens are indeed tumor-specific antigens. Another type of antigen, tumor antigens, are several types of overexpressed cellular proteins. As the name implies, overexpression, higher expression. We have higher amount of these antigens on the membrane of cells. We have two types of overexpressed antigens. First category are the genes that are silenced in normal cells. You can see the examples in the next slide. Another one are the product of genes that in the normal cells we have lower amount of them, but in cancer cells we have higher amount of them. Let me show you the slides. For example, cancer testis antigen. Cancer testis antigens are antigens that are in normal situation. They have normal expression in normal cells, but only in gametes and trophoblasts, not other cells. Clear? We have normal expression of a protein which is called several types of cancer testis antigens have been identified recently, and they are used as a tumor marker in the clinic. So we have a specific kind of antigen which is expressed normally on the normal gametes and trophoblasts. But in some situations like cancers, some type of cancers, we have the expression of these antigens on the cancer. For example, liver cancer, gastric cancer, colon cancer, and some other types of cancer. Several types of cancer testis antigens have been identified. We do not have them in the normal cells, in the cancerous cells, I mean. But in cancer cells, as a result of mutation, everything is a result of mutation. As a result of mutation, in cancerous cells, for example, in a stomach cancer cell, we will have sometimes expression of one cancer testis antigen. That it is not normal for cancer. Again, immune system can discriminate between cancer and normal cells. Another type of overexpressed cellular proteins are proteins that I told you are expressed in higher amount in cancerous cells. And HER2 is a growth factor receptor. It is a well-known tumor marker which is used for diagnosis of and for the detection of different stages of the different cancers, gastric cancers, breast cancer, prostate cancers. And besides, several types of immunotherapy now inside the clinic are being used against HER2. For example, Herceptin is a drug, is a medication which is used in the clinic against HER2. So it is a normal growth factor receptor. It is expressed normally on the surface of several cells. What's the matter?
It's
It
very
is
cold.
expressed.
It's very cold here.
No,
So you can shut down the.
Doctor, right?
Yes, I'll give you rest. After one or two other slides. Please. Here. HER2 is expressed normally on the normal cells in lower level. It's a growth factor. We have several types of growth factor receptor that they have normal physiologic role in the cells. They are growth factor receptors. But in some cancer types, for example for HER2, we have overexpression of this cytokine. So it is called overexpressed cellular proteins. You can see here that we have, for example, 3 epitopes of the HER2 on the normal cell, but in cancer cells we have more than that. There is differences in the amount of the antigen on the surface of the cells. What is going on with this?
الدكتور yang ketimbang.
So, we have a lower amount of the HER2, for example, here on the normal cell, but in cancer cell, we have higher amount of this. Do you remember one factor affecting immunogenicity of antigen was the dose of antigen? Here we have lower dose of the antigen, but this one in cancer, we have higher dose of the antigen. And differences in the dose of the antigen can affect the immunogenicity, induction of immune response. So whenever we have lower dose, there is no immune response. There is no immunogenicity. Whenever we have higher dose of the antigen, higher expression of the antigen, we will have immunogenicity, immunogeneration, or immune response induction.
So the protein that expressed on HC1, that only the protein that had a mutation?
Here I'm not talking about, yes, the result is mutation, but the mutation has changed the expression level of the antigen. Mainly this type of mutations are in the promoter of the gene. We have some types of mutation in the promoter of the gene that increase the expression level of the antigen. So we have mutation, of course, but this mutation hasn't changed the shape of the antigen. It has changed the amount of the expression of the antigen.
The normal protein expressed on MSC1 or not?
Yes, there is an important concept in immunology. In all normal cells, do you remember? In all normal cells, nucleated normal cells, we have MHC class 1 that they are presenting all intracellular antigens.
Okay.
All of them. Question?
No.
Another type of tumor antigens, please, are oncofetal antigens. Oncofetal, onco from oncogene, cancer, and fetal from fetus.
Yes.
They are normally expressed on the fetus, and they have a normal physiologic role in the fetus. We do not have their expression after birth. Is that clear? We have several types of genes that only are expressed in the fetus, but not in normal cells of after birth. In some situations like cancer cells, we have resume in the expression of this kind of gene. For example, and they are used as a tumor marker in the clinical laboratory for the monitoring of the treatment and for the detection of the tumor. For example, CEA, carcinogenic antigen, is expressed in the fetus. Or another example, alpha fetoprotein, another oncofetal antigen, which is normally is expressed in the yolk sac and liver of fetus, but we do not have their expression in normal cells, unless in cancer cells and sometimes in inflammatory conditions. So there is again differences between normal and abnormal cells. They are called oncofetal. There are several types of oncofetal antigens again that now are being detected in the clinical laboratory. Okay. Have a break for 10 minutes. Okay. You have questions?